Our current research interests are directed towards repairing the human spinal cord injuries, in which essentially the neurological problems are produced by axotomy. As you can see in this image, the brain is disconnected from the spinal cord after we have a cervical spinal cord injury. In this case, the brain is not capable of transmitting the signals to produce movement to the rest of the body, and the sensations cannot be conveyed to the brain, and in this manner we cannot feel the external world. To repair these injuries, we should implant some type of conductor to allow the axons to regrow across the lesion sites, to reconnect in the distal part of the spinal cord, and so restore movement. Presently we have two research lines. One is the field of motor control and the physiopathology of motor disorders after spinal cord injury. In this case, we develop different types of models of spinal cord injury in animals to reproduce the pathology of human lesions and to assess in which manner the organism deals with the lesion and how it compensates to recover the lost function. This has allowed us to measure with a high precision the type of motor disorders that are produced. In the case of cervical spinal cord injury, we have been able to assess how the arm works after the injury and how the body compensates the lost function. In this manner, we will be able to evaluate the beneficial effects of the different strategies that are being developed to promote functional recovery after these injuries. In our second research line, we work with the aim of producing some type of electrobiological device to implant in the lesion site. This type of device will contain conducting polymers, which are highly sophisticated materials that may allow us to apply electrical stimulation and simultaneously to present molecular cues to the neurons to induce axonal regrowth or to regulate or modulate glial cell responses. So we have two important findings. One is that we developed some type of new polymer that allow us to induce either axonal regrowth or glial cell proliferation and migration. Glial cells are very important because they are more numerous than neurons in the central nervous system, and they might induce by themselves axonal growth. Probably they will be very helpful to deal with the fibrotic response and to protect neurons after the spinal cord injury. This type of glial response is induced by growth factors that are presented in the material surface. In the other case, we are capable of functionalizing the polymer surface with highly specific molecules to induce axonal growth directly. These are cell adhesion molecules, and we hope that with the combination of both molecules that induce axonal regrowth and molecules that promote glial cell proliferation and migration, we will create a synergy to produce axonal regeneration in vivo across the lesion sites. Well, this type of electrobiological device is a highly complex challenge, and then it requires tight collaboration with other research groups and with the enterprises, not only in Spain, but across the world. We have collaborated mainly with the European organisms mainly with British, German, Portuguese and Greek partners in the research field. And presently we have focused in a critical point, which is having available the technology that is required to manipulate microfibers and micro nanotechnology to produce the devices that we need. In this sense, we are focusing in collaborating with enterprises, mainly in Germany and in the Basque Country, in Spain, because they have a very strong capability in the field of nano and micro fabrication, which is needed to deal with this type of problem.